We just finished talking about descriptive research and the three types, as you recall, are naturalistic observation, case study, and surveys. And calculating a correlation coefficient is how we describe the variables that we have observed in the descriptive research. And it is going to describe the relationship between the variables and how those variables change together. So that was our first category of research. Now our second category is called experimental research. And the experimental research method is used to demonstrate cause and effect relationships between changes in one variable and the effect that this produces on another variable. An experiment involves deliberately varying one factor and measuring changes in a second factor. When we conduct an experiment, we need to understand the two types of variables. We have an independent variable, and this is the variable that you manipulate or that you change. And this is also the variable that you hypothesize to cause an effect or a change on another variable. And this other variable is called the dependent variable. And the dependent variable, again, is hypothesized to be influenced by the independent variable. The independent variable must have at least two levels. You can have two categories, for example, a democratic versus an authoritarian class, for example, if we're studying the best way to teach college students. Another way is to design an experiment where you have an experimental group and a control group. And in this experiment, the experimental group gets the treatment and the control group does not. And this way you compare each of the groups to one another, or you compare the experimental group against the control group. Samples and randomization. Random samples, also known as random selection, is where each member of the population being studied has an equal chance of being selected for the study. For example, if I wanted to study the way college students at Front Range Community College study for a Psychology 101 exam, I might put every student who's enrolled in Psychology 101, put their name in a hat, shake up the hat, and randomly pull out 100 students. So the sample is the 100 students, and they are representing the population at large, which is all of the Psychology 101 students. Random assignment, however, is where every subject in the study has an equal chance of being placed in either the experiment or in the experimental rather, or the control group. So for example, perhaps a scientist thinks that he has discovered a great new treatment for depression. And he's done a random selection of his study participants. And now he needs to decide who's going to get his new depression drug and who's not going to. So he might put everybody's name in a hat that is part of the study, and then randomly select who's going to be in the experimental group, who's going to be in the control group. So remember, randomization helps avoid false results, and it gives us a representative sample, or a sample that closely parallels the population on relevant characteristics. And that allows us to extrapolate from the sample to the population in terms of the results. We can say that whatever we find with this small group of, say, 100 students, it applies to all of the Psychology 101 students because our sample represents the population as a whole. So randomization is an essential element to good scientific design. Sometimes we might have sources of bias in our experiments, and we need to understand what these are. So we can have what we call expectancy effects, and this is where a change in the dependent variable is produced by the subject's expectancy that that change should happen. So for example, if in that study that I just talked about with the new depression drug, if the subject knows that they are receiving the drug and not a placebo or sugar pill, for example, they may expect that drug to work. And the change in their depression, which would be the dependent variable, is not necessarily produced by the drug, but it might be produced by the expectation that the drug is going to work. So that's expectancy effects. We also may have a confounding variable in terms of demand characteristics. So these are subtle cues or signals that the researcher offers that communicate the type of responses that are expected. Here's what I mean by that. So say the scientist that developed this new depression drug is so excited about it, and he thinks he's going to get really rich. So when he passes out the depression drug to the subjects, he smiles. He might not be aware of it, but he might communicate some sort of cue that this is going to work. And when he passes out the sugar pill to the other subjects, he doesn't smile. Okay. So in this way, the researcher is communicating some sort of an expectation, and this can confound our results. So how do we control for sources of bias when we design an experiment? One thing we can do is make sure that we have a placebo control group, and this is where half of our subjects are exposed to, again, that fake independent variable, a sugar pill, that looks just like that new depression drug. Okay, and the effects of these are compared to the group receiving the actual depression drug or independent variable. So that's one way to control bias, where the subjects don't know what they're receiving, the real drug or the fake drug. However, that doesn't control for those demand characteristics that I just mentioned earlier, where the researcher signals some sort of an expectation to the subjects. So to control for that, we're going to do a double-blind study. And in this technique, neither the experimenter nor the participant knows which participant is getting assigned to the real drug and which participant is getting assigned to the placebo. So here we might have somebody in the lab that is separating the real drug from the placebo, putting them in numbered vials. So the person who hands out the drugs doesn't know what's in the vial because they are not labeled by the product. They're labeled by a random number. So this is where neither the experimenter nor the participant knows who's getting which treatment. It's called a double-blind study. Let's talk about some of the limits of the experimental design. Experimental design is often criticized for having too little to do with actual behavior because of the control that is infused in the laboratory environment. In other words, it's too far removed from the real world. So that's the first criticism. The second criticism is that there may be ethical considerations in creating real-life situations. By that, what I'd like you to do is follow this YouTube video link, and this is on the Stanley Milgram experiment. Go ahead and follow this link, look at this video, and I want you to remember that because of current ethical standards, this study could not be done today. So by now, I've assumed that you've watched the Milgram experiment, and let's talk about some ethical guidelines. The first thing is that we need to provide informed consent to the participants. They need to be aware of the general purpose of the study, and they need to be willing volunteers. They cannot be forced into participating. Students are often participants in psychological research because much research is done on college and university campuses. Can you think of any way where this might present an ethical conflict? Now, what I'd like to ask you is it ethical to ever deceive subjects in participating in an experiment? The answer to this is that we may not deceive a participant in general. If alternatives, however, are not possible, there may be an opportunity to offer some deception, but the good has to significantly outweigh the bad. Another aspect of the ethical guidelines in conducting an experiment is that we must protect the confidentiality of the participants in the study. And then finally, we must debrief the subjects after they have completed the study and let them know what the purpose was. This concludes my presentation for the scientific method.